On the appointed day, Herod, wearing his royal robes, sat on his throne and delivered a public address to the people. They shouted, "This is the voice of a god, not of a man." Immediately, because Herod did not give praise to God, an angel of the Lord struck him down, and he was eaten by worms and died. Well, welcome to Notable Nobels, a podcast about the Nobel Prize in Physiology or Medicine. My name is Harrison Doolin. I'm a postdoctoral scientist at the Fred Hutch Cancer Center in Seattle, and I will be your host for this web series. The purpose of this series is to trace key advancements made in the biological and medical sciences over the past 125 years or so, and we're using the Nobel Prizes in Physiology or Medicine as a guide. Now, every career has its highest prize, and for scientists, that prize is the Nobel Prize. It's the most prestigious award a scientist can receive, and it marks discoveries that have made a profound impact on our understanding of biology and our ability to treat diseases. So far on this podcast, we've been looking at Nobel Prizes awarded for discoveries related to infectious diseases. However, one class of infectious diseases has gone unmentioned. As you may have guessed from the quote I just read from the Bible's book of Acts, today we will be talking about parasitic worm infections. Specifically, we will be examining one half of the 2015 Nobel Prize in Physiology or Medicine, which was awarded to William C. Campbell and Satoshi Ōmura. The Nobel Assembly at the Karolinska Institute chose to give Campbell and Satoshi the award, quote, for their discoveries concerning a novel therapy against infections caused by roundworm parasites, unquote. We will be going over some descriptions of parasitic worms, and specifically the worm called Onchocerciasis volvulus that causes the disease river blindness. And we'll talk about the progress that has been made in eradicating this worm with the help of a drug called ivermectin. But first, let's cover the background of our Nobel laureates. William C. Campbell was born in Londonderry, Northern Ireland in 1930. He was raised in Donegal County, Ireland by parents who deeply valued education. He attended boarding school at Campbell College in Belfast, Northern Ireland, where he developed a liking for biology and an interest in becoming a veterinarian. He entered Trinity College at Dublin University in 1948, where he struggled with chemistry and physics, but he excelled in the biological sciences. After finishing his undergrad training, he moved to the United States in 1953 to attend grad school at the University of Wisconsin. His doctoral program was in veterinary sciences and zoology, and his research focused on studying parasitic worm infections in livestock. After finishing his PhD in 1957, he obtained a position as a parasitologist at the Merck Institute for Therapeutic Research in New Jersey. His work primarily focused on treatments for parasitic infections. While initially he was working to find cures for parasitic diseases of livestock, he eventually transitioned to studying drugs used to treat human infections. Campbell stayed at Merck for 33 years, and it was there that he would make his Nobel Prize-winning discovery with Satoshi Ōmura. Satoshi Ōmura was born in the city of Nirasaki in the year 1935. His mother was an elementary school teacher, and his parents impressed upon him the value of education. He attended the University of Yamanashi, where he was an organic chemistry student and also a competitive cross-country skier. After finishing his undergrad education in 1958, he obtained a master's degree in chemistry from the Tokyo University of Science, and in 1968, he completed a PhD in pharmaceutical sciences from the University of Tokyo. He was particularly interested in the biochemistry of microorganisms. He was skilled at the isolation, purification, and structural determination of molecules from microorganisms, including molecules with special properties like antibiotics. In 1965, he joined the Kitasato Institute in Tokyo, now known as Kitasato University, where he continued to do microbiology research. In 1971, he took a sabbatical to visit the United States. He arranged a lecture tour at universities around the country with the hope of sharing his research and setting up collaborations with American scientists. A crucial connection was established after he met Dr. Max Tischler, the former director of Merck's Sharp and Dome Research Laboratories, who had recently relocated his lab to Wesleyan University in Connecticut. Satoshi worked in Tischler's lab for a little over a year as a visiting research professor, during which time he was able to meet the scientists at Merck. Satoshi returned to Kitasato University in 1973 to take on the role of director of the antibiotic laboratory. From there, he maintained and grew his crucial collaboration with Merck, and it was during his time at Kitasato University that he would make his Nobel Prize winning discovery with Campbell, specifically the discovery of an extremely effective drug against parasitic worm infections. So, because we haven't talked about them up until now, let's take a moment to talk about parasitic worms. What are they, and how are they different from other pathogens that make us sick? Well, parasitic worms are very different from things like viruses. Viruses are the smallest living things on the planet. Like all living things, viruses reproduce, they carry genetic material in the form of nucleic acids, and that genetic material is used to produce proteins. Additionally, the genetic material of viruses is susceptible to mutations just like any other living organism. Viruses are special, however, because unlike other pathogens, they cannot carry out basic cellular functions like protein production or energy production on their own. In fact, viruses are not even truly cells since they don't have the defining feature of a cell, the cell membrane. In contrast to this are pathogens like bacteria. Unlike viruses, bacteria are truly cells. They have a cell membrane that contains a DNA genome and organelles for protein synthesis and energy production. But bacteria are distinct from other cellular pathogens because bacteria are described by scientists as prokaryotic. Now, prokaryotic is a fancy word that means without a nucleus. Bacteria DNA is not contained in a nucleus, and this is the defining feature of bacteria life forms. Additionally, all known bacteria are single-celled organisms. Separate from the prokaryotic bacteria pathogens are the eukaryotic pathogens, which include things like parasitic worms. Unlike bacteria, eukaryotes have a DNA genome contained in a nucleus. Classically, eukaryotic organisms have been divided into 4 groups called kingdoms. These kingdoms are the protist kingdom, the plant kingdom, the fungal kingdom, and the animal kingdom. Maybe some of your middle school biology is coming back to you as you hear these words. The protist, fungal, and animal kingdoms all contain organisms that can infect humans. Fungal pathogens are widely understudied, though there are several that can cause serious lung infections, and it's really a shame that scientists don't devote more resources to studying fungal pathogens. However, as much as I would like to talk about them more, we won't say any more about them here. They aren't the subject of this podcast episode. Now, pathogens that belong to the protist and animal kingdoms are referred to as parasites. The main difference between protists and animal parasites is that protists are typically unicellular, while animal parasites like parasitic worms are multicellular. Examples of pathogenic protists include Plasmodium falciparum, the parasite that causes malaria, Toxoplasma gondii, which causes toxoplasmosis, and Giardia duodenalis, which causes giardia. So far on this podcast, we've talked a lot about malaria, as well as bacteria and viruses, but we haven't talked yet about parasites from the animal kingdom, also known as metazoans. These parasites, by definition, are multicellular organisms, and some of them can grow big enough to be seen with the naked eye. There are 2 types of metazoan parasites, ectoparasites and endoparasites. The ectoparasites typically only infect the skin. They include things like leeches, ticks, and fleas. The endoparasites, on the other hand, live inside their host, and the most prominent of the endoparasites are the helminths, which we more commonly refer to as parasitic worms. So, to summarize, parasitic worms are eukaryotic, multicellular, animal endoparasites. Phew. Our story today focuses on the parasitic worm Onchocerca volvulus, which is responsible for causing the disease onchocerciasis, also known as river blindness. River blindness, as the name implies, is a type of blindness that occurs primarily in people who live near rivers, go figure. Historically, the disease was found exclusively along rivers in Africa, but the disease was introduced to South America several hundred years ago with the transatlantic slave trade. The disease is closely associated with rivers because it is spread by black flies that lay their eggs in flowing water. Incidentally, the scientific name of the fly species that transmits river blindness is Simulium damnosum, which appropriately translates to the damned black fly. The black flies are vectors for the parasitic worm onchocerciasis. The flies become infected with the worms when the female flies take a blood meal from an infected human. The juvenile worms migrate from the fly's gut to the fly's mouth. Then when the fly takes another blood meal from a different person, the juvenile worms are deposited onto the person's skin. The worms burrow into the subcutaneous tissue, which is the deepest layer of your skin, and once there, the worms are sealed off into nodules, which are often visible as bumps just under the skin. Over the next 6 to 12 months, the worms mature into adults. The worms undergo massive growth as they mature. The larger female worms can grow to be over a foot long or up to 50 centimeters, which is crazy when you consider that the black flies that carry the juvenile worms are only a few millimeters in length, so this is a huge growth spurt for the worms. Once the worms mature, the male and female worms mate. The female worms carry the developing eggs until the eggs hatch and then give birth to infant worms, which are called microfilariae. An adult female worm can give birth to over 1,000 microfilariae every day. The microfilariae leave the nodules where the adults are, and they spread throughout the body. They primarily stay on the skin, but they can also enter the lymphatic system and sometimes other organs. The skin infection can cause itching and is uncomfortable, but the big problem occurs when microfilariae enter the eyes. If microfilariae worms get into a person's eye, it can trigger an immune response that can cause tissue damage and scarring of the cornea, that clear outer layer of the eye. Extensive damage to the cornea causes it to cloud over, preventing light from getting through the eye and causing blindness. Although causing blindness is what onchocerciasis parasitic worms are primarily known for, the worms themselves are actually not trying to get into the eye and infect the cornea. The microfilariae are primarily found on the skin because the skin is where the worms can get picked up by another black fly that bites the poor infected person. And the goal of the parasite, to anthropomorphize it a little bit, is to get to the next host and start the replication cycle over again. So the eye is not the goal, the skin is the goal, because after a black fly picks up a microfilariae during a blood meal, the microfilariae in the fly's gut will mature into juvenile worms, ready to start the infection cycle once more when the fly lands on another person. Now, historically, humans have known about onchocerciasis parasitic worms for a long time. The first identification of the worms by a European was in 1890 when a German doctor in Ghana surgically removed a nodule from an infected patient. In the 1920s and 30s, the transmission of the worms via black flies was discovered, and the first reports of blindness associated with the worms were published. However, it wasn't until the 1940s that the scale of the problem was appreciated. In Western and Central African villages, around 10% and sometimes as high as 30% of adults suffered vision impairment or blindness because of the worms. Additionally, 50 to 60% of populations were found to be infected with the parasite and at risk of developing blindness. So we're talking millions and millions of people here. So how could doctors and scientists begin to take on such a daunting challenge? Well, initially, there was very little they could do. No therapeutic intervention existed, so surgical removal of the nodules that were visible was the main form of treatment. The removal of nodules could get rid of many but not all of the breeding females, but surgery did not remove the microfilariae, which are too small to be seen by the surgeon. Microfilariae could persist for years after surgery and still cause disease. And removing nodules from a patient surgically also did not mean they couldn't just become infected again when another black fly comes and bites the poor person. So, vector control became the preferred way to manage the disease. Starting in the late 1940s, scientists began using insecticides, primarily DDT, to eliminate black flies from rivers. This strategy helped reduce disease burdens, but it had two problems. The first problem was the negative environmental impact of dumping DDT into the rivers. And for more info on that, go back and listen to episode 12 of this podcast where we talked about DDT. And the second problem was that DDT was unable to completely eradicate the black fly populations. The flies tended to follow people around, including people traveling on boats, trains, or cars. So even if an area was wiped clean of the flies by DDT, the flies would eventually find their way back, either on their own or on the backs of humans. The persistence of the flies meant the persistence of the disease, and unfortunately for those already infected, there was little that could be done therapeutically to prevent the eventual progression of the disease to blindness. However, in the 1970s, a new therapy was introduced that would completely change the medical community's perspective on river blindness. That therapy would come from the work of our two Nobel laureates, Campbell and Satoshi. So how did Campbell and Satoshi develop an effective therapy for river blindness? Well, as we said earlier, Satoshi was working at Kitasato University and collaborating with Campbell at Merck. The partnership between the two institutions had an established workflow. Satoshi's group would work to identify new strains of bacteria that might produce useful chemicals like antibiotics or potentially any antiparasitics. Satoshi's group would test the drug candidates in cell culture, and any promising candidates would be sent to Merck for animal testing. Merck would then handle the large-scale production, marketing, and distribution of any successful drugs. When Satoshi began his work in the 1970s, he was particularly interested in the Streptomyces bacteria. These were the bacteria that several decades earlier had produced the remarkably successful and Nobel Prize-winning drug streptomycin. Satoshi believed that screening this group of bacteria might yield more highly useful antibiotic drugs. Streptomyces bacteria are found just about everywhere on the planet, particularly in soil, so finding samples of Streptomyces bacteria isn't all that hard. However, it's a tricky bacterium to grow in the lab. Satoshi, however, was a highly skilled microbiologist, and he set up his lab specifically to culture Streptomyces bacteria. Once his lab was set up, he began collecting soil samples to find new species of Streptomyces. He started with several thousand soil samples, but eventually narrowed it down to about 50 samples that grew well and were good candidates for testing antimicrobial drugs. He sent these samples off to Merck to see if any might be effective in animal models for a wide range of diseases. Over at Merck, Campbell was working as a parasitologist and was specifically interested in drugs that could treat parasitic worm infections. Campbell wasn't looking for drugs for human diseases like river blindness. He was a veterinarian and was interested in parasitic worm infections of livestock. But like most biomedical scientists, he started his experiments in mice. Campbell took extracts from Satoshi's Streptomyces samples and tested them to see if they had any efficacy in treating mice infected with intestinal roundworms. Most of the samples didn't help, but there was one extract that seemed to offer some protection. Campbell sent his results back to Satoshi, who decided to look at the positive sample in more detail. The promising Streptomyces species had come from a soil sample taken from a golf course in the Japanese city of Ito. This soil sample is arguably the world's most important golf divot, because after Campbell's positive mouse data, Satoshi and his team began working more intensively with this new strain of Streptomyces. Using modern methods of chemical separation and purification, they were able to identify the active molecule produced by the new Streptomyces species that was effective against worm infections. They called this new molecule ivermectin. The name ivermectin comes from the Latin word vermis, which means worm, and the prefix a, which means without, so literally without worms, which is an appropriate name for a drug that kills parasitic worms. Starting in 1975, Campbell began testing ivermectin in larger animals. Ivermectin was found to be an effective deworming drug in dogs and cattle, but the scientists didn't want to just stop with what nature had given them. They wanted to see if they could make an even more effective version of the ivermectin drug. Satoshi and his lab began chemically modifying ivermectin to see if any of the chemical derivatives might have greater activity against parasitic worms. One of the derivatives was given the name ivermectin. Ivermectin was found to be a safer and more potent version of the naturally occurring ivermectin. As an aside, this method is very similar to what Tu Youyou used when she was working with the drug artemisinin, which we talked about on our last podcast episode. Now, Campbell found ivermectin to be highly effective at eliminating parasitic roundworms in all kinds of animals, including sheep, horses, cattle, pigs, dogs, you name it. He published the first ivermectin animal results in the Journal of Medicinal Chemistry in 1980, and the following year, Merck began selling the drug for use in animals. For the next 20 years, ivermectin was the world's best-selling veterinary drug, making Merck a very pretty penny to be sure. But even before ivermectin became an agricultural wonder drug, Campbell was already speculating about its possible use in humans. In 1981, the same year Merck began selling ivermectin as a veterinary drug, a small clinical trial was conducted in Senegal, where a handful of patients infected with onchocerciasis parasitic worms were given various doses of ivermectin. The results were published in the journal Lancet in 1982. Among the patients that received the 2 highest doses of ivermectin, all of them saw huge reductions in microfilariae, and 6 of the 8 patients who received the highest dose saw complete elimination of microfilariae. Larger clinical trials with the drug were conducted in several African countries, and ivermectin continued to prove to be safe and effective. It consistently was able to completely clear microfilariae from infected patients. In 1987, ivermectin was approved for use in humans by the US FDA. The drug entered wide-scale use to great success and gained the attention of the Nobel Assembly. Thus, in 2015, Satoshi and Campbell were awarded the Nobel Prize in Physiology or Medicine. Once it became apparent that ivermectin and ivermectin were excellent parasitic drugs, one of the first questions for scientists was, well, how does the drug work? How exactly is it killing the worms? It was discovered that ivermectin is a neurotoxin. It binds to ion channels that prevent the worm's nerves from functioning properly, paralyzing and then killing the worms. More accurately, we should say ivermectin paralyzes and kills the infant worms, the microfilariae. Ivermectin doesn't kill the adult worms, the big worms that stay inside the nodules. On the one hand, eliminating the microfilariae is a huge benefit. Microfilariae are what are entering the eye to cause the blindness in the disease river blindness. So killing the worms before they can get to the eye saves people's eyesight. Additionally, eliminating the microfilariae prevents the black flies from picking up the worms when they bite an infected person, which prevents the parasite from spreading. Now, humans are the only known host species where Onchocerca volvulus replicates. So in theory, using ivermectin to prevent the spread of the parasites in the human population could eventually lead to the complete eradication of river blindness. On the other hand, this ideal scenario is complicated by the fact that ivermectin doesn't kill the adult worms, which means the female worms can just have more babies once the ivermectin is gone, repopulating the body with more microfilariae and putting the person again at risk for river blindness and maybe transmitting to other people. Now, the good news is that while ivermectin doesn't kill the adult worms, it does render the females temporarily sterile for many months after a single dose. In fact, ivermectin only needs to be given twice a year in order to keep an infected person from becoming repopulated with microfilariae. However, there's another problem. The female worm's lifespan. So the female worms can live upwards of 15 years. Now, that's a long time. So in principle, giving people ivermectin twice a year is enough to do 3 things. 1, prevent infected individuals from developing blindness. 2, prevent the spread of the parasite to new individuals. And 3, eventually cure an infected individual of the disease. But these benefits come with the logistical challenge of how do we get ivermectin year after year to those hundreds of millions of people at risk for developing river blindness? Remember, most of these people live in poor areas of the world and can't afford regular medical care. So what could be done to get these people the treatment they need? Well, to meet the challenge of river blindness, the Merck pharmaceutical company did something amazing. In October 1987, the same year ivermectin was approved by the FDA for human use, Merck announced that they would donate ivermectin for free to countries impacted by river blindness. The company pledged to donate however much ivermectin was needed for as long as it was needed until the disease is eradicated. Every year, Merck donates approximately 300 million doses of ivermectin that are distributed with the help of the World Health Organization and other public health entities. And this program has been going on continuously for nearly 37 years. Now, I know particularly in the United States, pharmaceutical companies get a bad reputation for putting profits over people's lives and are basically seen as greedy leeches. But Merck's ivermectin donation program is a nice contrast to that negative stereotype. We don't have to take it too far, though. Merck did make billions of dollars selling ivermectin as a veterinary drug, but on the other hand, they didn't have to give the drug away for free to treat river blindness, and they generously did so. So, however you want to look at it, there's no doubt that the campaign has been effective. Since the inception of Merck's donation program, 4 Latin American countries have been declared free of river blindness, and the disease prevalence in the Americas has been reduced to a small, difficult-to-reach area of the Amazon on the border between Brazil and Venezuela. Now, 99% of river blindness cases occur in Africa, and that continent too has seen great reductions in the incidence of river blindness. Thanks to ivermectin, as many as 1 million cases of blindness have been prevented. Another unexpected benefit has been the freeing up of agricultural land along rivers in Africa that previously had been neglected due to the presence of black flies carrying the worms. So ivermectin is having an indirect economic benefit as regions previously plagued by river blindness continue to eliminate the parasite. Thanks to the continued efforts of scientists, public health organizations, and local communities, it is possible that in the future, river blindness and onchocerca volvulus worms will be completely eradicated, and hopefully that time is coming soon. Okay, that concludes this episode of Notable Nobels. This episode was recorded on August 27th, 2024. I want to thank Digital Mind Productions for providing the music, and Nicholas Wynn for the graphic design. Next time, we'll again be looking at drug discovery, and specifically a new drug development strategy that differed sharply from the strategies we've talked about so far. It would have implications not just for infectious diseases, but for almost every single human disease you can think of. Want to know more? Well, listen next time to find out. Thanks so much for listening. See you then.